study our recent results. Hello everyone, my name is OHKI, coming from JAIST. Today I talk about three topics, and the first one is introduction. I talk about protein NMR a little bit. So I'm just afraid that this is some basic things. So I arranged this part for beginners, especially for students. So if you feel I know this kind of basic things, I give you 10 minutes reading time. So when I move to the next topic number 2, just I start our result presentation. In the second topics, I talk about new protein expression system developed in our group. And the last one, third one, is the application, NMR study of stomagen. Okay, let's move to introduction. Okay, NMR is nuclear magnetic resonance. NMR is one of the destructive analytical methods. So MRI used in hospital is a kind of NMR. And the NMR spectra provide valuable information of your sample. For example, structure, dynamics, interaction, and so on. So people in the NMR field believe NMR means no more research. That means once we get the NMR result, you don't need more experiment. NMR tells you everything. JAIST has 4 NMR machines from 400 MHz to 800. So these are photographs of 400 MHz NMR machine magnet, and this is big 800 MHz magnet. The height of 800 magnet is more than 2 meters, is almost 2.5 meters, and the weight is 3,000 kilograms. So superconducting magnet is cooled by liquid helium and liquid nitrogen. So why we need such kind of big magnet? Why we need strong magnet? Strong magnet means the higher frequency. So this is the carbon one-dimensional NMR spectrum recorded on 150 MHz. That means proton frequency is 600 MHz. And here is the carbon NMR spectrum at 100 MHz. That means the proton resonance frequency is 400 MHz. Which is better? Sorry, which is better? This one is much better than this one. Higher resolution, higher sensitivity, and looks so nice baseline. Okay. So this is one-dimensional proton NMR spectrum of sucrose. It's small molecule, quite nice spectra is observed on 800 MHz machine. So everybody can analyze this kind of spectra. So how about protein NMR? So this is a typical result of one-dimensional proton NMR spectrum of 16.7 kilodalton protein named calmodulin. Calmodulin is one of the famous calcium binding protein. We are using this protein for machine setting up. So as you can see, there are many peaks in this proton dimension. So do you want to analyze such kind of crowded spectra? No, I don't want to analyze this kind of spectrum. Too many overlap peaks. So to overcome this situation, how should we do? Normally we try this kind of two-dimensional proton NMR spectrum. This is a kind of proton NMR spectrum, noisy. Nose gives you the distance information of proton-proton pairs. Okay? There are many peaks in the off-diagonal region. So even for this region, okay, if this is methyl group region, we have still overlapping. So it is difficult to analyze this two-dimensional NMR spectrum. So there are too many peaks over too many overlap peaks again. So think about protein structure. This is protein backbone, NHCHCO, NHCHCO. Okay, this is one amino acid residues. Here is the side chain of residue I, and here is side chain of residue I plus 1. So now we are discussing only proton NMR. So there are many other atoms in protein structure, you know. For example, nitrogen is here, here, and carbon is here, 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 here. So let's move to text. Okay. So NMR can detect many spins. For example, the carbon. In nature, there are 3 kinds of stable isotopes for carbon. Carbon 12, carbon 13, and carbon 14. But NMR only can detect carbon 13. Okay. Then the carbon 13 natural abundance is quite low, 1.1%. So it is difficult to record NMR spectrum of carbon 13 for protein. So how should we do? This is the solution. Stable isotope labeling. When we make this kind of protein, okay, nitrogen 15 labeled with nitrogen 15. These are NMR active spins, and this is carbon 13, carbon 13, carbon 13, and carbon 13. So almost all spins are detected. Can we make it? We can make it using biotechnology methods. For example, genetically modified E. coli can generate your protein in liquid culture medium. When you produce your protein, you can buy carbon 13 labeled glucose and nitrogen 15, ammonium chloride. These are commercially available, so you can buy them and just dissolve them into the liquid culture medium. And then E. coli yield your sample protein using these carbon 13 and nitrogen 15. So we can get the labeled protein. And the NMR sample is very small, but the higher concentration is more than 0.2 millimolar, and we usually use 500 microliter small sample in the big magnet. Okay. Once we get the labeled protein, we can introduce heteronuclear axis like this. This is the HSQC spectrum to observe proton and carbon 13 correlation. It looks nice spectrum, and we also observe the proton and nitrogen 15 HSQC. So think about the protein structure. So each amino acid residue have one amide group in the protein backbone. So palmodulin is composed of 148 amino acid residues. So we see the quite nice separated 148 peaks in this spectrum. So one residue gives 1 peaks in this spectrum. So it is very useful spectrum to check the protein folding or protein interaction. And this is the recent standard methods, triple resonance 3-dimensional NMR spectrum. There there are many triple resonance experiments, and this is an example of 3D NMR, CBCA, CONH. These experiments give you the information, the correlation among them. C alpha carbon, C beta carbon is connected to the NH group of the proceeding residue. Okay. So this is the example of projection of proton and carbon-13 grains. So here is amide axis, here is aliphatic column. So each amide group have two peaks, C alpha and C beta. So the combination of three-dimensional NMR experiments gives you the whole correlation in the protein structure. So HNCA gives you correlation among this group, and CBCA and OG here, HNCA, CBC here and here. So overlapping is observed, just combine all data to analyze the speed. So there are many experiments, and we can use E. coli, you understand. But for example, E. coli is not good for the production of mammalian proteins. So how can we overcome? Let's move to the next topics. We developed the new protein expression system using plant cell. So there are many protein expression systems for stable isotope leveling. E. coli is the most major one, and PTEs, cell-free mammalian cells, insect cells, they are already reported, but nobody used plant cell for protein production for NMR sample. So we use it. So BY2 is one of the tobacco cells, plant cells. It's growing in liquid culture medium, and this BY2 lacks the chloroplast. That means that the cells do not do the photosynthesis, so they need carbon and nitrogen sources to survive. So we can use carbon-13 level stuff and nitrogen-15 level chemicals in the liquid medium, and then we get the proteins leveled with carbon and nitrogen-15. Okay, so this is a conventional method using BI2, but this is poor productivity. So we modified this system using RNA virus and its DNA, and the tomato mosaic wheel's DNA is tandemly conjugated to your protein's DNA, and RNA replication system makes your protein's RNA so much, and we get huge amount of protein. And X-ray is enhancer for this transcription, and estradiol is similar to IPTG in case of E. coli. So this is the result of production of GFP. Our system get a huge amount of GFP compared to the conventional method. And this system has another advantage, and this can produce mature protein like containing disulfide bonds, phosphorylated sample, and glycosylated sample, and so on. And this is an example of production of EPTI. EPTI is containing 3 pairs of 6 derivatives, disulfide bonds. This system produces the mature protein, so we don't need the refolding step. And this is HNCA strips. We can make carbon 13 and nitrogen 15 level BBTI. It's quite nice correlation from F45 to M52. Okay. This is another merit to use BICON system. We can produce amino acid type selectively labeled protein. So the upper panel, 4 panel indicate proton and carbon 13 entire HSQ2 spectrum, and these bottom 4 spectrum are ground. And I realized some students working on ground system, so I switched these topics to the front protein. Okay. So for this topic, I already published a paper in Nature Communication, and this article is open access. It means free of charge. So after my talk, if you are interested in, just access this site and download this paper and read it. Okay. The topic title is the NMR structure of stomagen reveals the basis of stomatal density regulation by plant peptide hormones. As you know, where stomata are pores on the surface of leaves, they exchange gas from plant body to atmosphere, or they get the CO2 from air. So for the long time, this is a big problem. How do lung plants regulate stomatal density? So in year 2005, it's 10 years ago, a Japanese group found the small peptide EPF1 and EPF2. The plant scientists overexpressed EPF1 and EPF2 in the plant body. The leaves doesn't have stoma. So EPF1 and EPF2 are negative regulators for stomatal development, they say. Then bioinformatician searched the similar protein in the Arabidopsis DNA genome sequence. Okay. And they found 9 similar peptides in the database. And the plant scientists checked the activity using knockout and knockdown plants, and they found this one increases the stomatal density. That's quite interesting thing. Okay. Normally, similar amino acid sequence had similar amino acid sequence had gives similar three-dimensional structure of protein, and then they show similar function, but this shows opposite effect on the stomatal density. This is a control. These are overdose experiment. We produce and purify the stomagen and EPF2 and overdose on the leaves, and just watch the surface of the leaves. There are many pores for this photo, but we have no pore in this photo. So stomagen increases the stomatal density, the vertical line, proportional to its concentration. And unlike this stomagen effect, EPF2 decreases the stomatal density. Okay. So we would like to know why and how. So we started the structure determination of stomagen using NMR. So it was difficult to prepare the stomagen NMR sample with E. coli because stomagen has 3 pairs of disulfide bonds. So it is difficult to produce E. coli by E. coli. So we use our new protein expression system with BICON, and we successfully get the NMR sample. And we tried many NMR experiments to determine protein structure, and we use many softwares to analyze them. And this is the result, one of the results of NMR spectrum, proton and nitrogen 15 HSQC. Quite nice dispersion, nice line shape. And this is another example of resonance assignment using 3D NMR. This is the combination of CDCS-COMH and HMCS-CB from isoleucine 33 to tyrosine 37. We see nice connectivity. Here's carbon axis, alpha carbon and beta carbon. Nice connectivities are observed. And this is important spectrum. NOESY gives you the distance information. The proton-proton pair is very close. This distance is within 5 angstrom. Okay. This is the downfield region of alpha proton, and here is schematic view of beta sheet. From 19 to 24 is one strand, and from 39 to 44 is another strand. These are forming the anti-parallel beta sheet. In this beta sheet, we see the very close proton-proton pair here, here, and here. Here is 24 alpha proton, and here is 39 alpha proton. Let's take a look. NOESY spectrum. So 24 is here in the diagonal peak, and 39 is here. So we see nice of diagonal peak, not NOE peaks here and here. So NMR data indicate these two protons are close. Okay, similar to this, we see nice cross peak between 22 and 41 here and here, and 20 and 43 here and here. So by using such distance information, we calculated the three-dimensional structure of stomach. This is the result. Okay. This is the overlaid representation of 20 structure of stomach. All heavy atoms are depicted. This is only backbone is depicted. 20 structures are overlaid. Okay. This is a real model of stomach. Here is N-terminal end, here is C-terminal end. Okay. Beta sheet is here, and one-term helix is here. These secondary structure components are connected by 3 pairs of disulfide bonds. Okay. This is results of a kind of dynamics experiment. The vertical line indicates the stability of each residue. So larger value indicate the region is stable, but the smaller region, smaller value indicate the flex. Okay. Sorry, doesn't work. I put the computer simulation results on here, but it can't work well. We try this one. No. Anyway, so these secondary structure regions are rigid, stable, and the loop region has flexibility. Of course, N-terminal unstructured region are also stable, unstable. Let's go back to the amino acid alignment of EBF family, and here is the loop region. Loop region shows this similarity, but the scaffold region has the well-conserved amino acid residues. So we tried the computer simulation and modeling of all family members, and we found all family members had similar three-dimensional structure. Here is stomach, and the loop region is totally different. So we thought the loop region is the functional side, probably. As the loop region has the biological function. To answer this question, what kind of experiment should we run in? So this is the best way to show the loop region is the functional side with or not. And this is stomatic. Here is the loop. Here is the scaffold. This is the positive regulator, and this is EPF2. This is negative regulator. Scaffold is here. Loop is here. So we designed and made this kind of mutant. In this mutant, the scaffold region is coming from EPF2 and loop region is coming from stomagen. So if this mutant shows positive function, so we can say the loop region is the functional side. So this is the result. So here is the amino sequence of the mutant. Red region is coming from stomagen loop, and green regions are coming from EPF2. This is result of the photo. Here is control. Here is the higher stomatal density. This density is proportional to the concentration. So we get the good results. It is enough to say the loop region is the functional side. It's not enough. We should try another experiment like this. So for this mutant, the scaffold region is coming from stomagen, and this loop is coming from EPF2. So we hope this another mutant shows the negative activity. So this is the result. We got the good result. This mutant shows the similar activity to EPF2, while the negative regulator. So we can say the loop region has the function. So what is the role of the scaffold? So we did this connection of these three disulfide bonds. We try to make a mutant CS, that means all cysteine residues are replaced with serine. So this CS mutant did not give any peaks in the downfield shifted proton NMR spectrum. So these are forming beta seed residues. So we say CS mutant doesn't have stable three-dimensional structures. So unstructured peptide doesn't show any activity in it. So the scaffold supports the functional loop. The folding is necessary to appear the function. And the last experiment is this one. Does the scaffold have any biological function? It's only supporting the loop. So we deleted this loop and connected these beta strands with short glycine linker. And this mutant has the similar fold to stomagen. It's well folded. The three-dimensional structure is very close to the stomagen. And here is the result of the overdose experiment. In all experiments, we use 3 micromolar stomagen, but scaffold concentration is increasing. With increasing scaffold concentration, we see decreasing the stomatal density. What does this mean? Scaffold has some biological function to recognize the target. We think the binding constant is very weak, but scaffold is necessary to identify and to recognize the target protein. So this is a summary of the structure-function relationship of stomachain. The blue converts the function, and here's the scaffold region. The scaffold region, consensus scaffold may have the role of molecular recognition. Interestingly, this region, molecular surface have very conserved cysteine residues. Cysteine cluster is here. So this slide shows the conclusion remarks of my talk. We developed a new protein expression system using BY2, and the new sample preparation technique provides a chance to study the new sample stomachain. And our results talked here indicate that structural biology is important for understanding the biological function at atomic resolution. So these are my collaborators and supported by these kind of grants. And thank you very much for your kind attention.
OHKI for delivering such a basic and fundamental research in NMR. I think it is a wonderful contribution because in NMR experiment we must need to have a good system for sample preparation. So I think his contribution is very significant. So now I would like to have few queries from the young minds if you have. Please don't hesitate to ask if you have something in your mind because the chance is very important for you. Or maybe you have chance in the breaks, in the lunch break, and we have tea breaks. These are the very, I think these instruments must be very expensive and may not be easily available everywhere. So in case if some of the students from this place wish to analyze their sample, do we have some facility in your center to analyze the sample and to make a chance for them?
So your question is for beginners?
Yeah, for if they can, if they have some interest to analyze their sample. So is there any possibility or opportunity for them to start collaboration or?
Just label with nitrogen 15 only and take proton nitrogen 15 HSPC spectrum and to look the peak dispersion and intensity and so on. And if yes, we go to the further NMR experiment, and beginners can start analysis of NMR spectrum using the free software on your own computer.
So I think there is ability for all of you if you wish to start, because NMR should be the extreme of the science. To know the ultimate structure analysis for biomolecules. Once you have, I think you can do wonderful science. So let's thank again to speaker Professor OHKI for making this invited talk.
So I want to ask that why can this NMR technique used for carbon 14?
Why this NMR technique used for carbon 14?
Why can it be used for carbon 14?
Well, time is limited. So do you know that quantum number and just try to go to the library and study physics. Okay. The keyword is spin quantum number.
You got it? Spin number. So these are the basic parameters that we use to analyze the atom. So okay, let me continue. Thank you again, Professor.